Genial. Pues hola y bienvenidos a todos a este nuevo seminario dentro del ciclo de seminarios de la SEMINARIO19. Nos disculpamos de antemano porque en enero pues no nos ha dado tiempo. Y bueno, pues hoy muchas gracias a todos por venir y quiero darle muchas gracias a Carolina Monzó, que nos acompaña hoy como ponente. Ella es bioinformática en el Instituto de Biología Integrativa de Sistemas en Valencia. Actualmente disfruta de una beca Marie Curie. Realizó su doctorado en el Max Planck, en el Instituto for Biology of Aging, y este trabajo pues, le ha servido también para ganar el acceso al primer premio Osvaldo Trelles de la SBBC. Así que tenemos mucha suerte de que esté aquí y que haya accedido a darnos un seminario sobre su tesis doctoral. Y pues sin más dilación le voy a dar paso. Como siempre os decimos que podéis hacer cualquier pregunta desde el mismo chat de Zoom, o si estáis en YouTube, pues también podéis hacer las preguntas que queráis por allí y se las transmitiremos a la ponente. Así que the floor is yours, Caro.
Muchas gracias. Gracias. Vale. ¿Veis bien, verdad?
Sí, perfecto. Adelante.
Vale. Pues nada, gracias, muchas gracias, Sara, por la presentación. Estoy encantada de estar aquí. Uy, no sé si me vais a ver. Ah, mira, sí. Genial. Well, I will do the presentation in English because usually it's easier. So I will first tell you about, well, my project was on the effect of dietary restriction on the microbiome and the adaptive immune system in mouse aging. And I will first tell you about our findings in the microbiome, and then I will talk about the adaptive immune system. So let's see. With old age, there is a higher prevalence of age-associated morbidities, such as cardiovascular disease, cancer, and dementia. And many studies have been trying to improve health at old age by improving the aging process itself. And in fact, several genetic, pharmacological, and dietary interventions have been shown to extend health and lifespan in different model organisms. Aging has been studied for many years, and among the hallmarks of aging, you see here dysbiosis is the dysregulation of the microbiome, and this is where we decided to focus my PhD. The microbiome is the collection of microbes that live in our bodies. It's sort of a forgotten organ. It includes 10 to 100 trillion microbial cells, primarily composed by gut bacteria, but there's also microbiome in the saliva and the skin and stuff like this. It's highly affected by age and diet, and it has many functions. Among them, there is energy homeostasis, absorption of nutrients, protection against pathogens, production of vitamins, and also regulation of the metabolism and the immune response. Hi. So also the microbiome generates metabolites that are essential for the host health. And what we know from bibliography is that in old age, the microbiome is losing diversity as well as the homeostasis in the bacterial population. So basically, you can see here that when we are adults, we have this very diverse and rich microbiome. And then as we are getting old, we lose diversity. There is a decrease in the good type of bacteria. And then these bacteria that are lost then are occupied by the less good type of bacteria, let's say. In some studies, in model organisms such as mice and killifish, we have been seeing that transferring a healthy microbiome to sick mice, like for example, in this case, has a beneficial impact on the health and the lifespan of the mice. And also transferring microbiome from young fish to old fish, like in this green case, it has a positive extension in lifespan. This could be due either to bacterial composition or to specific, like meaning the population, or to some specific bacteria. And we think that we can filter bacteria of interest by studying anti-aging interventions. And so, as I said before, the microbiome is highly affected by diet. So it would be logical that a diet-based anti-aging intervention would have a strong impact on the microbiome. Dietary restriction, here in red, it has been shown to extend lifespan by up to 40%. Dietary restriction, or DR, is the reduction in food intake by 40%, but without having a malnutrition. While, for example, ad libitum is the the common control normal eating. And so the problem is that these type of interventions in mice, they are usually done throughout their whole lifespan, but this is not really applicable to humans because I would really not like to be dieting my whole life to have a very healthy old age. And so in our lab, in the Max Planck, a few years before I arrived, there was a study in which they switched mice from ad libitum diet to dietary restriction at 24 months of age. And although they did not see a lifespan extension, they saw some positive effects on some biological markers. And so just before I started my PhD, a new project started in which they switched mice from ad libitum to dietary restriction at 16 months of age, which is like middle age for a mouse. And they saw that it was extending lifespan, almost recapitulating the lifespan of the mice that were chronically under diet. And then they also had a group of mice that only 4 months after were switched to DR, so instead of at 16, at 20 months of age. And in this case, they did not see this recapitulation of the extended lifespan, although some metabolic parameters were having a good response to it. So therefore, we thought that we could use these differences between the midlife switch and the late life switch as a filter to identify molecular aspects that would be associated with longevity. So with this information in mind, we defined the first two aims of my thesis. The first was to perform an in-depth longitudinal analysis of the murine fecal microbiome. And the second was to study the effect of aging, which would be this control ad libitum diet mice, dietary restriction, and the difference between the midlife and the late onset DR on longitudinal fecal microbiome and metabolome. We hypothesized that there could be changes in the microbiome and therefore in the microbial metabolome after late onset DR that would only recapitulate the DR-like patterns when DR onset was occurring like as late as 16 months, because we're saying like, this one is extending lifespan, but starting DR later in life is not. So we studied our two control groups, which were ad libitum and DR, and then also these two switch time points. We collected fecal samples from 12 months of age every 2 months until they died, and we were using 50 mice for each of the groups. So in total, we ended up having about 2,000 samples from 200 mice, and this was a longitudinal measurement. And this allowed us to do like a very high resolution longitudinal study of the microbiome and the microbial metabolome from each individual fecal, like original mouse. And we did this by sequencing the 16S subunit of the bacterial ribosome and performing mass spectrometry to identify polar and non-polar metabolites. So it has been shown before that the bacterial population distribution becomes dysregulated with age, but the population structure of the microbiome in dietary restriction remains stable. We corroborated this result in our dataset using linear mixed models, as well as, sorry. Okay, it's not here. Sorry. We also saw that the ad libitum and DR microbiomes were very distinct. We evaluated this using standard differential abundance analysis, and also we used some linear mixed effect, ridge regressions and elastic nets to try to predict the DR age of the mice using a model that we had been training on ad libitum mice. This means that since basically the microbiome of our DR mice, of an old DR, was not being classified as a young ad libitum. The diets were making the microbiomes very, very distinct. And so we asked ourselves if the microbiome of the midlife or late onset DR mice was adapting to DR. We tested this in many different ways. You can see here that we were using principal response curves in which we were comparing the microbial population to the baseline ad libitum, which is the blue one. And we saw that the DR mice in red were always having a different microbiome, while the 16-month switch in green was having like this almost recapitulation of the DR-like pattern. However, as we observed in the lifespan, we were seeing that the 20-month switch was not recapitulating these improvements, and we saw this as well in the case of the microbiome with the principal response curves. Then again, we saw that very few bacteria, when we look at which bacteria are different from ad libitum after introducing the DR diet, so if they have some sort of response, we saw that very few bacteria from the later switch were having a response, while there were more bacteria that were responding to the new diet in the younger switch. And then when we looked closer into which bacteria were different between ad libitum and DR, which are these red bubbles, and which bacteria were different between the late onset DR groups and DR, so bacteria that were not recapitulating these DR-like abundances. The number of bacteria resisting this possible adaptation to DR was higher in the case of the later switch compared to the younger switch. And thus we concluded that the ability of the microbiome to adapt to a DR-like pattern is high, but it declines with the age of DR onset. Knowing this, we looked for which bacteria could be associated with differences in longevity. We first looked into the overlap of those bacteria that were not adapting to DR-like levels in any of the late onset DR groups, because these bacteria that would be DR specific, so they could be associated with a slightly longer lifespan observed in chronic DR when compared to the younger switch. And as you can see, we identified 14 bacteria, most of them highly abundant in DR mice, and also some of them were even reported in human centenarians, for example, this Christenella here. Next, we looked into this group here. So these were which bacteria were adapting to DR-like levels in mice, starting at dietary restriction at 16 months, and then these were having this extension in lifespan. And they were not adapting to DR-like levels in mice that were switched to DR at 20 months, so no longer able to extend lifespan. We found 36 bacteria, some of them with lower abundance in DR mice than in ad libitum, others with high abundance in DR mice. And here it was important to see that most of the genera of bacteria that we found highly abundant in DR and in the 16-month switch were producers of short-chain fatty acid metabolites. And these metabolites can only be produced by the microbiome and are crucial in regulating the metabolism and immune responses. In some specific cases, such as these bacteria, simply by separating mice, regardless of which diet they were having, by the abundance of the bacteria, we could differentiate between which were going to be longer-lived and shorter-lived mice. So now that we knew which bacteria were associated with the differences between mice starting at DR at 16 and 20 months of age, as I said before, we also wanted to look into the metabolites, because the microbiome generates metabolites to communicate with the host. So we wondered how the microbiome changes we observed are reflected in the pool of fecal metabolites. And for that, we generated and target polar and non-polar metabolites from all these 2,000 samples. Basically, the polar are representing amino acids and the non-polar were mostly lipids. And we repeated the same analysis we did for the microbiome. In this case, what we learned was that while the non-polar metabolites are super switch resistant, like you see here in these two, that very few are reaching DR levels, the polar metabolites were showing quite a big difference in response to late onset DR. So the later case was way more resistant to the switch than the younger switch. So therefore, we zoomed into these polar metabolites because they were having this stronger difference between the two switches. And we asked which microbiome-produced metabolites could be associated with differences in longevity. So we again first overlapped the metabolites not adapting to DR-like levels in either the late onset or like both of the midlife and the late onset DR groups. These would be the DR-specific metabolites, and they could be involved in the healthy chronic DR phenotype. So it makes sense that first we check them out. And we found, for example, like adenosylmethionine or tryptamine, which have been previously associated with longevity. So in general, we were trusting our results. And then again, we focused here on the metabolites that were adapting to DR-like levels, especially in the 16-month switch, but not in the 20-month switch. And among them, we found different metabolites like gamma-glutamyl putrescine and tryptophan, which have been previously associated with longevity. But most interestingly, we found N-acetylspermidine, which is of special interest because it's within the pathway that generates spermidine, and it's a supplement that we use many times to extend lifespan in different model organisms. And thus, with this information, we confirmed our hypothesis that there are changes in the microbiome and the microbial metabolome after late onset DR that only recapitulate the DR-like patterns when DR onset occurs as late as 16 months of age. And in fact, now we have identified some of them. Finally, to wrap up this part of our study in the microbiome, we also compared the DR microbiome to that of mice that were subject to a genetic and pharmacological intervention, because as I said before, we could also use this to extend lifespan. And basically, in this case, we were using the reduction of the insulin pathway and the mTOR network, which are like super studied in the aging field. The first thing that we saw here was that the genetic and pharmacological interventions had a way milder effect on the microbiome. You see there are way less bacteria that are affected. But most importantly, we found some bacteria that were overlapping between all of these three interventions. There are these two that are most relevant for us because these could be contributors to the health benefits between these three long-lived models. And actually, this is making quite a lot of sense because the insulin pathway and the mTOR are both within the nutrient sensing pathways that are affected by dietary restriction. So in summary of this first part of our findings, we saw that dietary restriction ameliorates the age-related microbiome population changes. We saw that DR induces a distinct microbiome. The ability of the microbiome and the fecal metabolome to adapt to DR is high, but it declines with the age of DR onset. We also identified bacteria and metabolites associated with differences in longevity. And finally, we identified bacteria shared between longevity interventions. However, as I said before, we not only wanted to study the microbiome, but also its relationship with the host, especially with regards to the immune system. And what we know from bibliography is that there is increased dysfunction of innate and adaptive immune responses with age, and that in the process of immunosenescence, there is an increase in memory T cells and an associated loss of ability to respond to antigens and low-level inflammation. Within the adaptive immune system, the B cells are especially interesting as they can be directly binding into carbohydrates and lipids that are found in the microbial surfaces. And so B cells are involved in the control of the microbiome composition in its development and maturation. And they are also involved in, this age-related B cell function declines. They suffer from impaired protection against pathogens. Having low of this B cell function is also going to have a higher risk in infection and a decreased vaccination response. Maybe a good idea is to do a little bit of, maybe later, but a little bit of dietary restriction before getting a vaccination. But I will show you why I'm giving this idea. So in general, it seems like studying the host's B cell mediated immune responses could be crucial to understand the aging process. A little bit more context about the B cells. They are characterized by having this hypervariable membrane-bound B cell receptor, or BCR. And with age, it has been reported that there is a loss of the BCR repertoire diversity, as there are clonal expansions, which are after maturation, the B cell that has the same membrane-bound antibody copies itself many times. So and then this proliferation undergoes a little bit more diversification, but in general, you have very similar antibodies. There is also an increase between individual dissimilarity, and then there's also impaired primary and clonal B cell selection that in turn is going to have a negative effect on affinity maturation. In an easy way to understand how these four metrics are basically talking to each other is if we imagine that different individuals are different buckets. In the young age, these individuals have many different dots that would represent many different B cell receptors. So we have a lot of diversity. So by chance, young individuals are more similar to each other because there is a higher possibility of finding antibodies that are similar. But then in old age, there is this clonal expansion, and every different person is going to expand a different set of clones, which means that, and if you imagine that the amount of different B cells that you can have is up to 100%, and you are occupying 90% by having clonal expansions, then there is only 10% of the space left for there to be a little bit of diversity. And so we are super different from each other because we are choosing different clones to expand. So the question for us is, how does the B cell receptor repertoire change under dietary restriction, and whether the BCR contributes to the difference in longevity between the midlife and the late onset DR mice. To study the BCR, we decided to sequence the hypervariable region of the B cell receptor. This is the part that binds to antibodies, and you probably remember that the way the BTJ segment, this hypervariable part, is generated is by the recombination of one V, one D, and one J gene into a constant chain. That is what is going to tell us if it's IgA, IgG, AG, whatever. There is extra variability around this part, the CDR3 region, by inclusion of nucleotides. And then, as I was saying before, during maturation, when there is a clonally expanded set of B cells, there is a phenomenon called somatic mutation that I represented here with the asterisks, that is just going to give us a little bit extra diversity within the clones. So this rearrangement is about 600 base pairs, and therefore we could use Illumina sequencing in an asymmetric manner. So 400 times 100, but since we had ligated adapters to both sides, then we could do 400, 400 and recover the whole segment. And since this information, we wanted to get it to bridge this communication between the microbiome and the host. We started by looking into the B cell receptor repertoire of the gut, which is in direct contact with the microbiome, obviously. And then that of the spleen, which is a lymphoid organ that would give us a general overview of the systemic response of the B cells to the late onset dietary restriction. And so we collected these organs in mice that were 5 months old, so the young time point, and then old mice in 2 time points at 20 and 24 months of age. We sequenced their B cell receptor repertoires, and with this experimental design, we went in with the question of, does dietary restriction delay the age-related changes in the BCR that is found in old mice? And if there is an effect when the DR is initiated at 16 months of age. So first, we tested our hypothesis in the ileum, and we were quite surprised to see that dietary restriction had a very mild effect on the BCR on the ileum. Out of all the different characteristics of the BCR we evaluated, only the phenomenon of somatic hypermutation was affected by DR, which had a higher level of diversification by somatic hypermutation, but most importantly, neither the 16-month or the 20-month switch, you see them here, they are grouping with the ad libitum. Neither of them had an effect on somatic hypermutation. But this was a little bit surprising for us, because before, in our longitudinal study, we saw that there was some response of the microbiome always when we were starting the diet. And when we took these specific mice in which we were looking at the B cell receptor and we sequenced their microbiome, we saw that this characteristic general adaptation to the microbiome of the DR mice was also happening. And since this was puzzling us, this uncoupling of the microbiome and the B cell receptor repertoire, we went to look into the spleen, which was the organ that we had originally in mind. So in this case, we did see that dietary restriction slows the age-associated decline in the B cell receptor repertoire diversity. You see here, this red line is going down, but it's not at the same level as ad libitum. It's significantly different, meaning that the number of B cells there are, it's higher than in ad libitum. And also there is a slow, it's delayed this abundantly clonally expanded set of B cells in old age. You see them here, that the DR is separated from ad libitum. So not only that, but also we looked into the response of the switched mice at the midlife and the old life mice, and then these 2 metrics and also many others, the values of the 20-month switch were sometimes grouping with the DR mice and sometimes grouping with the ad libitum mice, while the 60-month switch, the one that was extending lifespan similar to DR, was grouping always with the DR mice. So this could highlight that this difference between the responsiveness of the B cell receptor repertoire between one set of mice and the other could have an impact on these lifespan differences between the groups. And so with this information, we were confirming our hypothesis that DR delays the age-related changes in the B cell receptor repertoire that have been reported in old mice. And also it has an effect when initiated at 16 months of age, which is telling us that the age of DR onset affects the responsiveness of the B cells of the adaptive immune system. So having observed how dietary restriction slowed the age-related decline in the B cell receptor repertoire diversity and this increase in clonal expansions that were generally slowed the age-related changes in the BCR, we were wondering whether these changes might be associated with the health state of the mice. And thus, this could contribute to the lifespan and health span benefits of both DR and the switch at 16 months. So we correlated all the metrics we studied in the BCR repertoire to a macromorbidity index that we designed that was basically containing the pathology. So when we were dissecting the mice, we were writing down if they had any pathological, like any pathology that was relevant, from like big spleen to kyphosis to blindness or whatever. And then on this value, we're also adding the impact of cancer, because cancer is a morbidity of age. We were scoring tumors as 0 would be the absence of tumors, 1 would be one tumor in an organ, and 2 would be 2 or more organs affected. So there would be like this value of metastasis, sort of. And as you can see here, the red ones are the ones that are significant after multiple testing correction. The high levels of clonal expansion here and the low levels of diversity that we had observed this in the libitum mice, the ones that were like having their shorter lifespan, were highly correlated with morbidity. So these metrics were also likely contributors to the increased lifespan and health span of DR and the 16-month switch mice. So just in summary of this last part, we concluded that the B cell receptor repertoire of the ileum is only mildly affected by age and by diet. Dietary restriction delays the age-associated changes in the splenic B cell receptor repertoire. DR initiated in midlife has beneficial effects on the splenic B cells of the adaptive immune system. And splenic B cell receptor repertoire features that are different between midlife and late onset DR mice are correlated with lifespan and health span differences. If you want to find out more about this B cell part, we published it a couple years ago, and you can find it here. And just to finish my presentation, I would like to say thank you to Linda Partridge's lab at the Max Planck, which is where we did the project, and everyone that was involved. The funding from back then, funding from now, and also my current lab and the SEBIPCE for inviting me to give their talk. So thank you very much. And if you have any questions, I will be happy to answer them.
Thank you very much, Carol. It's a huge work for a thesis. Yeah. So anyone has a question? Las podéis hacer en inglés o en español, que el
Sí.
inglés no os pare.
Es que la ciencia es más fácil contarla así, pero...
No, sin problema, digo por si acaso. Déjame comprobar. Es que como estoy solo ando cambiando de pestaña del YouTube al Zoom, vale, en YouTube no tenemos nada. Bueno, I have a couple of questions, so I can start. So maybe we can break the ice. Because in bioinformatics, I love methodology. I want to ask you something about that. How's been your experience integrating all these omics? Because you've analyzed microbiome, you've analyzed sequencing of the BCRs. How was your experience integrating all these analysis and this data?
Complicated. So, for example, for the metabolome and microbiome analysis in the longitudinal study, we had a lot of help, of course, from the facilities of the Max Planck. And the main thing was that there are databases that are telling us which metabolites are produced by which bacteria. And it's true that even though when we do metabolomics, we get thousands of compounds, naming them is very complicated. So maybe out of 10,000, you get 60 names. And usually you know which metabolites come from where. So it's very easy to just link it back manually, and it's not really a problem. Like you just generate the database and then just link it back. But for example, in the cross-sectional study, the one where we do the B cell receptor repertoire, if you remember in the ileum part, I showed you some microbiome data. And in that case, we did have to do some integration of the data because we wanted to, I don't know, like to build more of a multi-omic model. This was way more complicated. We were using tools like Mixomics, which is for like multi-block integration. And I'm not sure how much I learned from it, honestly. I think if you are integrating something like, I don't know, like DNA with RNA, with proteomics, with metabolites, sure. But in this case, looking into the B cell receptor repertoire and the microbiome is not going to tell me a lot.
Yeah, I think when I started 10 years ago in bioinformatics, like integration of omics was like that thing, but I think
Yeah.
we haven't learned anything
Yeah, I mean,
since then. Yeah.
now there are some other tools like there is More, for example, that is from a collaboration from my lab with another lab that is doing like these gene regulatory networks, bringing in different omics. There are others based on, I don't know, like flux balance analysis, and then you're integrating different types of omic modalities. But I honestly think that this is one field within the bioinfo community that is still needing a lot of work. And
Thinking
I
about
really
it,
struggled.
yeah, no, I think it makes sense because it's something that when you are integrating omics, they are too specific, the question. So it's difficult to have a tool for analyzing to answer all the questions you can ask to omics. So I think that's what bioinformatics are for, not right?
Yeah, yeah, but this is what you said. I think it's very important. Like you need to figure out what is the question that you want to ask to your data set and if it makes sense to integrate them. Because in my case, it didn't make sense to integrate the BCR with the microbiome. And although I did try, then I realized that it didn't make, like it wasn't going to give me anything relevant biologically. So I forgot this. But yeah, I think that having the biological question very clear is even more important. Like it would be the most relevant thing for me.
Yeah, you first need to think, make the question, and then try to ask
Yeah.
to answer. So, and another question also about methodology. I've never analyzed or seen data from BCR sequencing. What are you looking for? You are trying to you are looking for just the sequence. You are checking if there are mutations. How do you analyze that?
So I think it's very cool. Maybe in the micromorbidity index, you can see some of the features that we evaluated. So first of all, well, you get your normal FASTQ files.
Yeah.
So there are some tools that are going to help you reconstruct this, like because I was doing 400 plus 100, and then 400 was plus 100, but I had UMIs, so I could get back into which two 400s were going to give me the full sequence, and I could merge them together to get the full length of the
Full sequence.
of the BDJ rearrangement. And there are many things that you can look into. If there is an overrepresentation of one set of like one type of V gene or J gene, that is going to tell you something about the issues that you're having with recombination, if there's a skewness towards one set or the other, because there are many families within the V genes. You can also look into if like for example, the CDR3 that I was saying is this super hyper variable part within the rearrangement. There have been some studies that in age they were saying that the length of this, because you add nucleotides right around the D gene to bind it to the V and the J. So the length, if it's going to have an impact on aging or not. You can look into like these generalized metrics that I told you about, diversity, like clonal expansion. You will want to look into sequences that have the same rearrangement, V, D, and J, and they also have the same CDR3, but then they have mutations along the whole sequence, which means that after they were clonally expanded, they did this hypermutation, so they come from the same original B cell. Yeah, basically you can look into as many things as you want. And sometimes there are like full research projects that are based on whether this B family is over selected. In my case, I just wanted to have a general idea of how the BCR were responding to this anti-aging intervention, because there was, I think, like something similar done in fish, in like in fish that had been treated with an anti-aging intervention that was different from DR, and this was the first study in which the repertoire was being looked into, like, I think, with in a model organism that is closer to us. So yeah, I just looked at everything.
No, it's really cool because when you think about sequencing, like it's so difficult to sequence repetitive sites, and this is the sequence in the genome that is more repetitive and that has more rearrangement. So I can imagine it's crazy to analyze this kind of data.
Yeah, so luckily for me, I mean, if you look into the RNA, you just tag it with a UMI at the beginning, and then you can reconstruct this, and you're not going to have a problem because you don't have this repetitivity of V, 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 J, 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 J. It's only one rearrangement. So Illumina-wise, this is okay. If we had to do this like without rearranging on the DNA, we would have struggled a lot more. And then in those cases, you would need other types of technologies.
Okay, so just last question by me. How can this be? I think this is the most common question here, but how can these findings be transferred to humans once you? It's the big question, right?
Yes, of course. I think they are super transferable, and probably, I mean, I'm talking about dietary restriction, which is like the hardcore next level lifespan extension done in mice. So these mice eat very little, they are super thin. But I'm sure someone has heard about intermittent fasting. This is like the milder version of dietary restriction. So it is quite translatable to humans, I think. And there are studies that are already being done in the Netherlands and Germany in which they are having people doing dietary restriction, not to the level of a mouse, not to 40%, but like 20% maybe of reduced intake. And they have many positive impacts on like glucose tolerance, insulin tolerance, like heart rate. There are many parameters that I didn't show because like I was talking about the data analysis, but we also had all of the phenotyping of the mice that were positively impacted. And this is also being seen in humans. So I encourage everyone to try it. It's not terrible.
So eat less.
Yeah, or at least try this idea of the intermittent fasting. I think it's very interesting.
Yeah.
Also, in that case, you need to think about your circadian rhythm, how it's going to have an impact and stuff. But I think it's quite translatable. In the case of the insulin, the pharmacological and the genetic intervention, you're not going to modify genetically anyone. So this is out of the question, the insulin knockouts. But rapamycin is a drug that is used for treating like in cancer patients. And in the US, for example, they already started using it as an anti-aging intervention, that and metformin and other things. I think in general, it's translatable because it has a positive impact on your health, but we need to figure out dosages and like how to set this organized properly so that we are not hurting anyone. We are only getting the good stuff.
Yeah, I think it's pretty famous also, the intermittent fasting. So
yeah,
we have to eat less in summary. Okay, so I don't know, last opportunity to make a question. I think, let me check on YouTube. No. So in any case, you can reach Carolina at any time. I think she would be happy to answer you any questions. And thank you so much for accepting to being here today. And good luck with your career.
Thank you very much.
Bye, everyone. Ok, estamos para.
He puesto ahí y hablar rápido.